Hi, we are ProteinVision. We develop state-of-the-art models for various protein prediction tasks, and we produce videos that explain the most popular deep learning concepts and models in the field of biology. For various models, we provide a step-by-step explanation, plus a Python-based guide for how to use and train them. In this video, we cover the ESM-2, one of the most widely used protein language models for sequence-based predictions. So let's dive into the ESM-2 model. A protein consists of a sequence of 20 different amino acids, and on average proteins have a few hundred amino acids. When a protein is synthesized, it does not remain a linear sequence, but instead folds into a 3D structure. Each amino acid is a small molecule made of bonded atoms, shown here in different colors. Hence, the folded protein is a compact 3D structure made up of thousands of atoms. The 3D structure defines the function of a protein, but most of the information about the protein is already stored in the linear amino acid sequence. This is why the ESM-2 model simply uses linear protein amino acid sequences as model input. The model architecture of the ESM-2 is a standard transformer network encoder. ESM-2 is a series of models in different sizes. We will first focus on the most commonly used ESM-2 model with 650 million learnable parameters. This model consists of 33 encoder layers, where each layer consists of a multi-head attention function with 20 attention heads and a feed-forward neural network. The token embedding dimension D is 1280, and the hidden dimension in the feed-forward function is 5120, which is 4 times the hidden dimension D, a typical ratio in transformer networks. We're now going to look at some implementation details, and start by looking at how the input embeddings are computed. In a transformer network, we need to divide the input sequence into smaller subsections, the so-called tokens. For proteins, each amino acid is a separate token. The input embeddings assign to each token a unique vector that encodes the type of token. The dimension of the input vector is equal to the hidden dimension D of the network, 1280. The set of all possible tokens in the model is called the vocabulary. ESM-2 has a vocabulary size of 33. 25 tokens encode amino acids, the standard 20 amino acids plus 2 non-standard amino acids, as well as tokens for ambiguous amino acid letter codes. Additionally, we have 8 special tokens, such as unknown, classification, beginning of sequence, and end of sequence token. The token embeddings for all 33 possible tokens are stored in an embedding matrix of dimension 1280 times 33, which is the hidden dimension D times the vocabulary size. Each token is mapped to a token index, and when inputting a token sequence, we can simply access the columns corresponding to the token indices. In the ESM-2 model, the embedding matrix is learned during training. We also need a way to encode not only the type of the amino acid, but also their positions within the sequence. Typically, we would obtain position embeddings for each position in the input sequence in a very similar way as I've shown you for token embeddings. These can be learnable or fixed if we use sinusoidal positional encoding. We would add the position vectors to the token embeddings, resulting in token representations that encode both the type and its position within the sequence. However, ESM-2 uses an alternative to these positional embeddings, the so-called rope embeddings. We are now going to look at how rope embeddings work, which is a popular positional encoding method that is, for example, also adopted in the llama models developed by Meta. ROPE stands for Rotary Positional Embeddings, and the broad idea is that instead of using some kind of positional embeddings that we add to the token embeddings, we rotate the token embeddings by a certain degree. And how much we rotate the token embeddings depends on the position. For example, the first position would result in only a small rotation, while later positions are rotated proportionally more. For simplicity, let us assume our embedding dimension is d equals 2. This means each token is represented by a 2-dimensional vector. The methionine amino acid at the first position in our sequence is displayed here as the yellow token embedding vector that does not store any positional information yet. To add positional information, we rotate this vector by an angle of 1 times theta. We multiply theta by 1 because this amino acid is at the first position in our sequence. For the methionine at the fourth position, we would rotate the same methionine token embedding vector by an angle of 4 theta. We can also visualize this for vectors encoding two different amino acids. If we assume the yellow vector represents the amino acid methionine at the first position and the blue vector alanine at the third position, we now need to rotate the yellow vector by an angle of 1 theta and the blue vector by an angle of 3 theta. When computing the attention scores in a transformer network, we compute scalar products between the rotated vectors. Here I name the yellow vector v and the blue vector k. Scalar products only care for the length of the vectors and for the angle between them, which means whenever there is a distance of 2 between the tokens in the linear sequence, we get the same outcome for the scalar product of their embeddings. This means we only encode the relative position between amino acids in the sequence, and we do not encode their absolute positions. Intuitively, this means that the model learns to understand the order of tokens by how much their embeddings have been rotated, instead of relying on added positional information. And tokens that are close to each other will be rotated by a similar degree, which indicates to the model that those tokens are close. We just had a look at how we rotate embeddings in 2 dimensions, where it is clear how we rotate a vector by an angle theta. However, in 3 dimensions, it is unclear how to choose rotation axes. So, to overcome this problem, if the dimension is larger than d equals 2, the rotation is applied to pairs of values inside the embedding vector. The first 2 entries are the first pair, entry 3 and 4, the second, and so on. The reason why we apply the rotation to pairs of 2 values is because rotating 2D pairs is simple and it preserves embedding structure. Each pair is rotated by an angle that depends both on the token's position m and on the angle theta k that depends on the specific dimension pair. Lower index dimensions rotate more slowly, while higher index dimensions rotate more quickly. This design has a nice effect. Slow rotations in the lower dimensions help the model recognize long-range patterns across a sequence, while fast rotations in the higher dimensions help it to focus on fine-grained local relationships. So how and when do we apply these rotations in a transformer network? In contrast to traditional embeddings, we do not apply them in the input layer. Rope embeddings are applied inside each encoder layer just before the attention scores are calculated. More specifically, they are applied to the query and key vectors, but not to the value vectors. This way, the positional information directly affects the computation of the attention scores, and thus how tokens attend to each other, but rope embeddings do not directly affect the token representations themselves. Rope embeddings have been found to improve the model's ability to generalize to longer sequences and to learn positional relationships more flexibly than traditional absolute positional embeddings. With rope embeddings, the model only cares about the distance between 2 tokens, and hence we can apply the resulting transformer network also to sequences that are longer than any training sequence. With learned absolute positional embeddings, this would not be possible. We will now look at the final architectural details of the ESM-2 model. In the feed-forward block, the ESM-2 models use a GELU activation function instead of a ReLU activation function. As you know, the ReLU function is 0 for negative values and leave positive values unchanged. The GELU function is very similar, but as you can see, it also returns small negative values for small negative input values, which means this function provides a smoother output compared to ReLU for values that are close to 0. This can help optimization by avoiding sudden gradient jumps, and the smooth behavior better preserves information through deep layers. Now, let us look at how the ESM-2 model was trained. We remember what our encoder does when we input a protein amino acid sequence. The sequence is divided into tokens, where each token is an amino acid of our input sequence. And the encoder then processes this sequence and outputs a numerical representation for each amino acid. To train the encoder in a self-supervised way, Randomly, roughly 15% of all amino acids in the input sequence are masked. This means, for example, that we do not let the model know what type of amino acid we have at the 4th position. How we do this is we add an additional token to our model, the so-called mask token. ESM-2 has a special masking token in its vocabulary that tells the model that we don't know what the amino acid is at that position. We then train the model to predict what type of amino acid we have at the masked position. To do this, we can train a single layer neural network on top of the corresponding token representation, which outputs a vector with 25 entries, where each entry corresponds to one amino acid. For example, the first entry might correspond to the amino acid alanine, the second to K, which represents lysine, and the last one to methionine. For each amino acid, we get a score between 0 and 1 that indicates how likely it is that we have a certain amino acid at that position, where values closer to 1 indicate that it is more likely that we have this amino acid at that position. In our case, we would input the vector at the 4th position, and we would then hopefully get a high score for methionine, which is actually the true amino acid at that position. So, how do we train the model to predict the correct amino acid for the masked positions? For all the masked positions, we can create a binary vector with all zeros and a single one indicating what the true amino acid is at that position. We then compare the vector y, storing the correct amino acid, which is the vector that we want to predict, with the vector y hat containing the predictions of our encoder using the cross-entropy loss. This training approach is called self-supervised training because the model uses input data itself to generate the data for the training task, and it does not require any external data to be labeled or measured. But why is this self-supervised training task, where we randomly mask 15% of the amino acids in the input sequence, a good approach to generate better and more meaningful representations of the amino acids in our sequence? To solve this task of predicting the type of a masked amino acid, the model must extract information from the other amino acids in the sequence. We remember that the linear sequence folds into a 3D structure, and hence not only the amino acids that are close in the linear sequence, but also in the 3D structure are important to guess the type of the masked amino acids. The model thus learns the spatial context in which the amino acids are located, and it also learns the dependencies between amino acids, which is critical to understanding protein structure and function. Although ESM-2 was trained only to generate useful representations for masked tokens that we can then use for predicting the type of amino acid, to accomplish this task, the model must also generate informative representations of the other tokens so that we can easily extract information from them when we try to gather useful information about the masked token. This means that if after training we apply a protein amino acid sequence that is not masked to the trained encoder, we still get useful and informative representations of all the amino acids in the protein sequence. And these representations not only store information about the amino acid itself and its position in the sequence, but also about its context in the sequence and its dependencies with other amino acids. We can now compute the element-wise mean of all amino acid embeddings to obtain a single protein representation. Alternatively, we can obtain protein representations by adding a classification token and fine-tuning a pre-trained ESM-2 model to predict the property of the proteins, for example, the pH value under which the protein functions optimally. After this fine-tuning, we can extract the updated classification token, circled in green at the top corner of the screen, and we can use it as a task-specific protein embedding. Finally, we look at different existing ESM-2 models. 6 different ESM-2 models were published, ranging from 8 million parameters to 15 billion parameters. In this table, you can find for each model the number of parameters, the number of layers, the hidden dimension, and the number of attention heads in each multi-head attention layer. Typically, larger models work slightly better, but also require higher computational resources. You should now have a good understanding of how the ESM-2 models work. In the next video, which you can access via the video description, we will look at the Python code for using the pre-trained ESM-2 models to generate protein embeddings, as well as fine-tuning the models for specific downstream prediction tasks. If you enjoy our videos, please like and subscribe.